All right, DNA replication. We have our DNA, tightly coiled DNA. We have up here our first strand. The top strand is our leading strand. The bottom strand here is our lagging strand. Now, like I said before, that this DNA is tightly coiled, we need to loosen that up just a bit. And an enzyme that can do that is topoisomerase. Now, to help even further with this unwinding, we have helicase, another enzyme. Now you see here at the replication fork that the two strands are separated. And in order for them to stay separated, we have stabilizing proteins. Our next enzyme for discussion is DNA polymerase. So this is DNA polymerase. Specifically, 1, 2, 3. DNA polymerase 3. Now, DNA polymerase 3 moves down the leading strand and synthesizes 5' to 3'. All right. Now, how does the DNA polymerase 3 know where to start? That is a very good question. Well, we have our RNA polymerase primase that creates primers that act as, like a starter, a starting place for DNA polymerase 3 to begin synthesizing. Once DNA polymerase 3 begins its synthesize, starts here at the primer and begins synthesizing, then there's no need for the primer. So DNA polymerase, let's erase this, will then take the primers away or remove the primers, and then it will replace that gap with nucleotides. So that's where you see here a nice, clean, complementary strand. So we're going to change this. And we see our complementary strand. All right, on to our lagging strand here at the bottom. A lot goes into play with this. So remember I mentioned that we have primers, that there's one primer that begins on the leading strand where the DNA polymerase 3 begins to synthesize 5' to 3'. Well, the same thing happens here on the lagging strand. Primase, RNA polymerase, will then place primers at various spots on the lagging strand. So you'll see more, let's see right here, more frequently primers placed on the lagging strand versus the leading strand. So we have our enzyme primase, which is an RNA polymerase. And they place down primers or starters, our starting spot. Our DNA polymerase 3, let's put it right here. DNA polymerase 3 will begin synthesizing 3' to 5'. So DNA poly 3 will synthesize 5' to 3'. And we see that. So now we have a primer, then we have a segment of DNA, then primer, and then a segment of DNA. So now that we have our primers and a segment of DNA, we need to kick out the primers because that's not part of the DNA. And polymerase, DNA polymerase. So DNA polymerase 1 will then remove primers and place nucleotides or DNA in its spot. Nucleotides in its spot. So now we have a gap. Then we have DNA, then gap, and then DNA. But that gap needs to be replaced with nucleotides, and that's what DNA polymerase 2 does. Now, the segments of DNA that I mentioned earlier, just a few seconds ago, are called okazaki fragments. Oops. So they are short segments of DNA on the lagging strand. Now, once we have our nucleotides, our complementary DNA on the lagging strand, they need to be glued together. And the enzyme that helps with that is called ligase, DNA ligase. I'll just put it down here, DNA ligase. And it acts as glue. And that is our DNA replication. Need more help? Click the link in my description box to get started with your microbiology study module.